Hi everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder why it is so hard to remember where you put your keys, or why your memory seems to work differently when you are a kid compared to when you are an adult? Well, today we're diving into some fascinating research that looks at how our brains learn and remember, and how that changes as we grow up. This new study, titled "The Memory We Miss," really dives into how we measure memory, especially in developing brains. Think about it. We often test memory by seeing if an animal or a person prefers exploring something new over something old. That sounds simple enough, right? But what if this standard way of testing misses some really important details about how memory actually works? Especially in young animals, that is the big question these researchers are asking. So, in many animal studies, especially with rats, scientists look at things like how much time an animal spends sniffing or touching a new object versus an old one. If it spends more time with the new one, we assume it remembers the old one and prefers the novel. This is super useful, but the paper argues that by only looking at this one measure, we might be missing out on a whole lot of other interesting behaviors that tell us even more about how memory develops. They even draw a parallel to studies on fear, where just looking at one behavior like freezing and not other behaviors like darting can give you a limited picture. The core idea here is that how an animal explores an object might tell us just as much, if not more, about its memory as how long it spends with it. They're suggesting a richer way to look at behavior, going beyond just "did they remember?" to "how did they remember?" They're especially interested in what happens during development. The researchers explain that the common tests like novel object recognition, or NOR, and novel object location, or NOL, rely on a built-in preference that rodents have for new things. This is a great starting point, and it has helped us learn a lot about the brain areas involved in memory. These tasks are also good because they don't require things like food rewards or electric shocks, which can sometimes interfere with studying natural learning processes. The authors point out that even though these tests have been valuable, they can be a bit too simple. They suggest we need more standardized ways of measuring behavior. And importantly, we need to be open to looking at a wider range of behaviors. They highlight that just looking at how long an animal interacts with an object might not capture the full story, especially when comparing young animals to adults. The paper then goes into some detail about the brain circuits involved in memory for adult rats. We're talking about areas like the perihinal cortex, which is important for recognizing objects, and the hippocampus, which is crucial for remembering locations and longer-term memories. They explain how these areas work together, but they quickly pivot to the developmental aspect. This is where things get really interesting for the study. They explain that while we know a good deal about adult memory circuits, we know less about how these circuits develop. This is where behavior becomes a really important tool. By observing how young animals behave differently, we can start to piece together how their memory systems are maturing. Here is a key point. The traditional measures like discrimination ratios might actually hide important differences. They use the example of fear conditioning again. Scientists used to just measure freezing as a sign of fear, but then they discovered darting and active escape behavior. What they found is that females were more likely to dart, and this was linked to different brain activity. This showed that by only looking at freezing, they were missing about half the story, and that different brain pathways might be used by males and females. This is the core argument they're building. If we can find similar hidden behaviors in memory tests, we can get a much deeper understanding of how memory develops. The researchers then talk about how the objects themselves matter. They bring up the idea of affordances, which is a concept from ecological psychology. Basically, an object's properties, like its shape, texture, or whether you can climb on it, afford certain interactions. A chair affords sitting, a ball affords rolling. They argue that scientists studying recognition memory often don't pay enough attention to these affordances. Different objects can lead to different behaviors and potentially different types of learning. And this might be especially true for developing animals. The way an object encourages interaction, like climbing or sniffing different parts, could be a key to understanding how an animal learns. This is where they present their own research, and it is pretty cool. They tested young rats, around 21 days old, pre-weanlings, and adult rats. They used two types of 3D printed objects in a spatial recognition task. NOL. One object was flat and asymmetrical, with a clear top and sides that were easy to climb or sit on. They called this the flat asym object. The other was pointed and symmetrical, which was harder to climb and didn't have as many clear orientation cues. Now, if they had just looked at the standard discrimination ratio, basically how much time they spent sniffing the novel object versus the familiar one, they would have concluded that both young and adult rats remembered equally well for the flat asym object. They showed good memory for that one. For the pointed asym object, neither age group showed much memory. So, based on the usual measure, It would seem like there is no big developmental difference here, but here is the twist. When the researchers looked more closely at how the rats were exploring, they found something amazing. Adult rats, regardless of the object, mostly spent their time standing up in the open arena, or rearing young rats though, showed a fascinating shift when the objects were the climbable flat asim ones. The young rats spent a lot more time sitting on or climbing on the objects. But when presented with the pointed TSIM objects, they switched their behavior and spent more time standing and looking around the open arena. This is the memory we miss. The young rats weren't just passively exploring. They were actively changing their behavior based on the object's affordances, and it seemed linked to the type of memory they were using. In the spatial task, climbing on the object might actually help them build a better spatial map. The adults didn't show this same kind of shift in behavior. This suggests that young brains might use different strategies to learn, and these strategies are influenced by the environment and the objects within it. Even more intriguingly, when they used a task that tested object identity, remembering the object itself, not its location. With these same young rats, they didn't show this behavioral switching. They behaved more like the adults. This is crucial, because it shows that the complex exploratory behaviors observed in the young rats were specific to the spatial memory task, suggesting a more sophisticated developmental process than just general exploration. The researchers proposed that these different exploratory behaviors, like climbing and rearing, might be actually helping the young rats build their spatial memory systems. They mentioned that spatial memory relies on things like place cells in the hippocampus, and these systems are still developing in young rats, so perhaps interacting with objects in a more complex way by climbing on them provides extra spatial information that helps fine-tune their developing brain maps. This is a really elegant idea. The way young rats interact with objects isn't just random behavior. It might be a crucial part of their learning process, a way they actively engage with and build their understanding of the world. For adults, their memory systems are more mature, so they might not need these elaborate strategies. So, what is the big takeaway? The paper argues that we need to move beyond simply looking at discrimination ratios and start analyzing the full spectrum of behaviors. This means that two animals might get the same score on a memory test, but they could have reached that score in completely different ways, using different learning strategies and possibly different brain mechanisms. If we only look at the final score, we miss these vital developmental differences. The authors are calling for a more nuanced approach to studying memory. Instead of asking, did they remember, we should be asking, how did they remember? What in the environment helped them learn? And which brain parts were involved at different ages? They believe that by looking at the rich tapestry of behavior, we can unlock a much deeper understanding of how memory develops. The paper also includes a table that shows just how varied the methods are, even for standard recognition tasks. This highlights the need for more standardization, but also for embracing new ways to analyze behavior. The figures and tables in this paper really help visualize the experiments and the different behaviors they observed. We can see the types of objects and how the young rats changed their behavior depending on whether they could climb or if the object had clear spatial cues. So, what is my take on this research? I think it is incredibly important. It challenges us to rethink how we study memory, especially in developmental contexts. By focusing only on simple metrics, we risk missing out on the complex and fascinating ways that young brains learn and adapt. The concept of affordances is powerful, reminding us that the environment and the objects within it are not just passive backdrops, but active participants in shaping our cognitive processes. The strength of this study lies in its detailed behavioral analysis and its focus on developmental differences. They didn't just settle for the standard measure, they dug deeper and found a really interesting behavioral split in young rats that adults didn't show. This suggests that the strategies animals use to learn spatial information might change significantly as they mature. However, like all research, there are limitations. This study was done with rats, so directly applying these findings to human memory development needs caution. Also, while they observed interesting behavioral shifts, directly linking these specific behaviors to specific neural circuits in the young animals requires even more in-depth neurobiological investigation. But that is exactly what makes this so exciting. What does this mean for us? Well, it suggests that the way children explore, play with objects, and interact with their environment might be fundamentally building their understanding of the world and their memory capabilities. It reinforces the idea that play is not just fun, but a crucial learning tool. For adults, it makes you wonder if we too, in subtle ways, might use different strategies to remember things depending on the context and the nature of the information. Future research could build on this by exploring these detailed behavioral analyses in other developmental stages and in different species, including humans. Imagine designing toys or learning environments that leverage these developmental differences in exploration to enhance learning. It also opens doors to looking at how individual differences in exploration might relate to different memory profiles. This research is a fantastic reminder that understanding the brain and memory is a journey, and sometimes the most profound insights are found when we look beyond the obvious measures and explore the subtle, yet significant, ways that behavior reveals the inner workings of the mind. Thank you so much for joining me here at RUBIN'S Mind Lab. If you found this breakdown helpful, please give this video a like, leave a comment below with your thoughts on memory development, and don't forget to subscribe for more explorations into the fascinating world of psychology. See you next time.